Hello and welcome to this special show on Rajya Sabha television. I am Vishal Dahiya and here we take a stock of the battle against COVID-19 pandemic. Now, one of the very important aspects in this fight against this particular pandemic is the mutations in the SARS-CoV-2 virus and how those mutations will affect our response to not only the virus in terms of battling it, but also specifically looking at the vaccine which is being developed. We'll look at all these aspects. We'll try and understand more. And for this, we're joined by a distinguished panel of guests. Let me first introduce them to you, beginning with, we have Professor S.S. Vasan with us. He's Dangerous Pathogens leader at CSIRO Australia. He's the one who has led a study on this aspect as well out there, which we'll be talking about in a little while. We also have with us Professor N.K. Ganguly, former Director General of Indian Council of Medical Research, and we also have Dr. Vishwakarma, advisor to CSIR. Welcome all of you gentlemen to Rajya Sabha television. Let me begin with you, Professor Vasan, and let's begin with by trying to understand a bit more about the mutations as far as SARS-CoV-2 virus is concerned, and specifically the G strain.
Thank you, Vishalji. The SARS-CoV-2 virus, as you know, is an RNA virus, and viruses mutate. There is nothing alarming about that. However, we do have to keep track of these mutations. In the last few months, we have been tracking tens of mutations in the spike protein of this virus. And it was flagged sometime in late April by our colleagues in the United States that there is this new strain called G strain. So the virus which emerged from China, that is the older D strain. This new G strain is now replacing the older D strain, and it has become the dominant version of the virus. So if you look back in early May, this strain was present in India in about 50% of the sequences, and in the world at that time it was in 2/3. Then 2 months later, when we looked at it, India had gone up to 2/3. The world level, it had gone up to 75%. And now you can see on the screen, it's actually, at the moment, 85% of the sequences in the world are this G strain. And in India, the value is 82%. So we obviously have to look at the impact of this mutation, what it means in particular for vaccines.
Okay, and what exactly would you say out there based on your study, Professor Vasan, in terms of the effect these kind of mutations does have on the efficacy of the vaccine candidates which are being worked on right now?
So there are 321 vaccine candidates in development. Most of them target the spike protein. So it is important for us to understand whether this mutation will affect the vaccines. So when we studied this in my laboratory at the CSIRO, we found that actually this G strain is not going to be affecting the vaccine efficacy. So the vaccines were developed with the older D strain genome sequences, by and large, and we tested them against both the old strain as well as the new one. Mm-hmm. We found that the vaccines work equally well.
And why do you say that?
We say that because we tested it using the sera from vaccinated ferrets, and we were able to show good neutralization, comparable neutralization with both the D strain and the G strain. Mm-hmm. And this is really important because most of the vaccines are targeting the spike protein. So there are other mutations which we are also studying, but this is a very encouraging result because it would be problematic. Like, for example, with the flu virus, seasonal flu, as you know, the virus constantly mutates. So every year you have to tweak the vaccine. We call it vaccine matching, one for Northern Hemisphere and one for Southern Hemisphere. Based on what we know up to this point, we don't believe that's going to be a big problem for the COVID vaccine.
Okay. Let me bring this to other experts on the panel out there. Professor Ganguly, how would you view the mutations as far as SARS-CoV-2 virus are concerned? We'll talk about the vaccine part later, but the mutations which are happening.
Yeah, in India, we have both strains from Wuhan, from China, which was G strain, as well as from Europe, which was a slightly different strain. So there are several mutations. Large mutations are happening and small mutations are happening. In India, when it began, it was A3I, which was the major mutation. At the moment, it is A2A. This is a major mutation. And then there are minor mutations. However, the G, GH, GR, L, V, S, O, all clades are there in India, more or less in the different proportion.
Okay.
And there are about six laboratories in India who are doing the NGS through Novasig, and they are mapping how it is spread around the country. At the moment, majority of them, as Sheshadri said, they are around spike protein. And one of the things which is done when you develop vaccine, that you test them not only against ferret or monkey neutralizing antibodies, you also test them with the plasma, convalescent plasma antibodies.
Mm-hmm.
There are about 600 different kinds of antibodies which develop in the convalescent plasma, monoclonal clones. And
Mm-hmm.
when you do that, and when it reacts to them appropriately, you at the moment you are safe that this mutation at the moment will not affect. In the future, what will happen, nobody can say. In influenza viruses, mutations happened in a way that every 2 years or 3 years you have to change the strains. However, in India, there is a scientist whose name is, someday you can bring him on your show. He is in the Indian Institute of Science. His name is Dr. Vardarajan. Dr. Vardarajan has opened a company known as Minavax, and he is working on a futuristic vaccine construct which will not be affected through mutations. But mutations tell many important things. It not only tells the origin and how it is spreading in India, like in Iceland, which depends on tourism. Every tourist who descends there, two of his samples are taken and sequenced completely. So they are creating a tremendous map there, and so that if they in future they need something, it will happen. We have another Indian scientist whose name is Srinivasan Ramani.
Mm-hmm.
He is in Oregon. So when they have sequenced it, there of course in US it is D614G plus. There is the main mutant over there. When he has seen that, they have been able to really do much more than others, that they have been able to say that this person will go from outpatient to inpatients, this person will go to ventilator or ICU. They
Okay.
have also been able to track the infection. So in the tracking, they have been very, very successful, and they have been able to track and create clusters which are much better than the hotspots which are done through normal surveillance. So it's
Okay.
a very strong molecular surveillance. As a molecular surveillance tool, it is also very, very important. But this database, if it is kept, it will help in our future vaccine designs. But
Okay.
some of the designs which we need to follow, which will actually not be affected by mutation. So in influenza viruses also, now the designs which are being made, they are being made of the proteins which will not be affected by mutations. So every year you will not have to change.
Okay, okay. Professor Ram Vishwakarma, your initial views on those aspects which both Dr. Vasan and Professor Ganguly are bringing out in terms of Indian context as well when we talk about efficacy of vaccine, because we have two vaccine candidates being worked on indigenously as well.
Yeah, so answer is that we don't know as yet. These are very early days, and total so far, if I am correct, 90,000 viral genomes have been sequenced worldwide, and total 12,000 mutations have been reported.
Mm-hmm.
And the main mutation, which is worldwide, is the common which Dr. Shashadri was telling, is the main D versus G. G, because of its open structure, now we have three-dimensional structure of this mutated proteins, and that has a faster entry through ACE2 receptor, so it spread faster. But the correlation of these mutations with vaccine efficacy is really not known as yet. And we should also look at the other side of the mutations, that all mutations are not really bad. There may be some mutations in virus which destroy the virus itself against the immune system. So there are both kind of mutations. There's one kind of mutation which I'm worried from therapeutic point of view, the mutation in RNA-dependent RNA polymerase gene, which is actually causing remdesivir resistant. There is a likelihood of having remdesivir resistant. So I think when we look at these mutations, not only from vaccine point of view, we look at from 3 point of view: the therapeutic monoclonal antibodies that are in development, the therapeutic small molecules that are in development, and obviously the vaccine in development. But these are very early days. The best good news about this virus is that it's very slow mutating virus compared to influenza virus or HIV virus and other viruses. And every 2-3 months you have 1 or 2 reported. So it's a really very reasonably stable virus. So I think there is nothing to be alarmed with. There is no sort of impact on efficacy of any vaccine that are in development. So I think we should take it as a biological process. These viruses mutate, like Dr. Shahadri said, and that's part of the life cycle. And some of the mutation actually will become someday Achilles heel of the virus itself, and it will destroy the virus itself against the immune system.
Okay. Dr. Seshadri, do you agree with what Professor Vishwakarma is pointing out there? We know a little less right now, and your study sheds more light on it, but not much to worry about in terms of the efficacy of vaccine.
That's absolutely right. And I think we are in agreement, all of us, that this is a virus which has a proofreading mechanism. We call it exoribonuclease. What this means is, compared to influenza viruses, it won't mutate that fast. And also the things that we are tracking at the moment at the CSIRO, and I'm pleased to say we actually collaborate with Dr. Vinod Skaria at the CSIR in Delhi. And we've jointly produced this site called COVID Beacon. And we are actually looking at the mutations, not only from India or Australia, but across the world. There are, as you heard, thousands of mutations. But when it comes to mutations which have consequence, both structurally and functionally, we are probably looking at this stage, we are tracking around a few tens of mutations which we think might become problematic. We also need to bear in mind mutations can accumulate. So we need to keep that under tracking, which is exactly what we are doing. So as you heard from our esteemed colleagues, this is not going to be a major problem, but it's one thing to say it's not going to be a problem. The paper we published in the Nature Vaccine is the first experimental proof backed by computer modeling that actually we can reasonably say that. And that's very important, in fact.
Okay.
And as you also heard from Dr. Vishwakarma, you will find that this mutations results in the virus over a long time when the COVID-19 spreads. Once you have fewer and fewer susceptible people, you will find that the virus will be selected naturally for those what we call clades, which won't necessarily give you a very severe disease or a mortal disease, but which will linger much longer in your system until it can find some other host, some other person to infect. We actually call it the survival of the flattest.
Okay. Well, but one more aspect, Dr. Seshadri, here, and the point which has been raised by Dr. Vishwakarma, in terms of the effect of the mutation vis-a-vis therapeutic aspect of it, in terms of monoclonal antibodies there as well. Is that seemingly another cause of concern? Leaving aside the vaccine part.
Not yet, but that's what scientists around the world. There is incredible knowledge sharing between all the countries that are affected. And in particular, I'm pleased to say, between Australia and India. And we have to together divide and conquer this workload, because there are so many mutations. All of us can't study all of them. So it's really important that the mutations that are emerging to be of consequence, not only for vaccines, but also for therapies, as we heard, we need to be tracking them.
Okay. Professor Ganguly, your views on those aspects vis-a-vis a therapeutic ways intervention specifically, if you look at it from the mutation point of view and the concerns out there specifically? We agree that in terms of vaccine development, it might not be that problematic, but on the therapeutic part.
Yeah, actually, Vishal, mutations and drug resistance is an age-old thing. It is not new. And this is how we actually identify that these mutations are related to the drug resistance. At this given moment, previously we were not really paying that much attention to minor mutations. But now, with the NGS and very strong bioinformatic tools, you get trends in the mutations which more definitively help towards finding out the clinical responses also. Previously, there could be mutation, but it may not relate clinically. But now you can find it very, very accurately. And every mutation changes some function in the virus or the organism. And in that way, when a drug becomes non-functional, you may have to design it differently so that it becomes functional. So this particular thing of all those people who develop drug, they go on doing it. But one of the major significance which is coming up, which I'm tracking, that we are now finding that with these mutations, some of the mutations are inconsequential. They will disappear or they will not survive. But some of the mutations will survive, and some of them will also form in the future reassortants. They will combine and they will become reassortants, and some of them will establish and start circulating. That will be one of the dangerous signals. It has been seen in HIV, it has been seen in many. So these mutations in the United States, some of these mutations have started in the dendrogram, they have started to separate. And they have started becoming distinct from the other ones which are circulating there. And United States also, both European and Chinese strains have established themselves. So there the infection is so dense that these mutations are now happening. So we have to track the mutations and ultimately create a database where we will identify the function. But functions are already being identified. One major function which is being identified now, that we know that this will be more virulent and this might be less virulent. And we also can now, with the mutations, actually 3 major groups they are doing there, one in the Phoenix, one in the Oregon, and one in the UCSF, that they are trying to predict through a dense analysis that whether this person will go to mechanical ventilator or this person will not go to mechanical ventilator. And with a great accuracy, they are going to predict it. So sequencing and mutations, and we have seen this. We won the polio war because we did a molecular surveillance in polio, and India was in the forefront of this. So India has now established several major biobanks with these strains, and at least 6 laboratories which I know of, they are doing the NGS and sequencing and creating database. What we need to do now, as Sheshadri said, and quoted Vinod Skaria's work which they are sharing with IGIB, when I said that A2A is now established in India, this is Vinod Skaria's work. And actually, he has published this now. This is an published realm. That is why I quoted this. So this is his work only. So the main challenge in India will be data sharing with the international group, because previously data was not being shared. It happened when H1N1 California CDC published it right the next day when the sequences were known. Or the Ebola Zaire, 90 Zaire strains sequences were published in the public domain. And Hugo now says that sequences you can't patent. So this actually controls the ethical part of the organic sequences, the genomic sequences. So we need to share them so that we know that how they are moving in the countries, how they are establishing themselves, and how the character of the disease is changing. It is also changing with the population. There are host parasite interactions and host immunologic interactions. Being immunologist, I know that this is an immunologic disease of inflammation rather than the virus. So all of these are very, very important, a very important database, and I'm very happy that Indian scientists are doing quite well. And in this particular arena, Indian scientists are very good. And some very novel vaccine designs are being brought by Indian scientists based on these sequences.
Okay, definitely. The more we know about the virus, the more easier it will be for us to understand how it behaves and then bring in, measures to control and prevent the further infection as well. Dr. Ram Vishwakarma, your views on the way forward here, from what we know, what we need to know, as well as putting it in, in implementation phase when we talk about making drugs or vaccines against this particular virus.
Yeah, so my view is that there is nothing to be alarmed of. These mutations will be discovered, and these mutations are in genome of the virus. But so long as that genome information is not transferred to structural proteins, I'm not worried about. So 2/3 of these mutations means nothing as far as their information going to protein is concerned. So this is all very nice for scientists to create that knowledge, but in immunological science and therapeutic science, it really doesn't impact. Second part, I would say, most of the vaccines, when you give, it's not one set of antibodies that have been generated. There are set of antibodies with several clonal sort of histories of B cells, and then you have the T cells involved in that. So it's a very composite response by immune system to the virus. In fact, our understanding towards immune system is very rudimentary compared to what we are learning about virus, I think. So this is a very, very evolutionary interplay that is happening, and I would say that there is nothing to worry about, and it will help more knowledge we have, better we are in terms of designing next generation therapeutics, next generation antibodies, and also for the vaccine. So it's an evolving process. We are in a very early preliminary phase of this understanding, I would say this. So I think any guess or any prediction based on what we know so far is very, very hazardous. I would say this as a scientist.
Okay. Professor Vasan, your concluding comments before we bring this discussion to an end?
I entirely agree. I think we have to be careful in saying, based on what we know up to this point, we are not alarmed. We are reassured. The news from my team, which looked at the impact of this D614G mutation, is that vaccines are unlikely to be affected, which is great news for over 321 vaccines in development at the moment. The other point I would link back to Ganguly Saab is that when you are having these virus genome sequences, it's very good to have the associated patient metadata. What we mean by that is what happened to that patient. Did the patient survive? Did the patient have a severe disease or a mild or moderate disease? What are some of the risk factors for the patient? For example, do they have hypertension, or do they have diabetes? Do they smoke? We don't need to know the identity of the patient. We don't need to know their name or where they live, etc. But actually knowing what their age is, whether they are male or female, or any risk factors, what we call comorbidities, these would be really useful, because you can then use the information in a more holistic manner. So what we were talking about, Dr. Skaria's work at CSIR in India, collaborating with CSIRO in Australia, one of the things that we are now trying to do is to have that associated patient information, but without the identity of the patient, so that we can correlate whether this is happening because of the virus or is it happening because of the host. And this is what Professor Ganguly was saying. The host pathogen interaction is really important. And a lot of efforts are going on, and I'm very pleased at the level of collaboration that is happening at the moment worldwide, and in particular with Indian experts who are absolutely at the forefront of science.
Okay, there it is. Thank you so much, Professor Vasan, Professor Ganguly, as well as Dr. Ram Vishwakarma, for sharing your views with us and our viewers on this important aspect. As our panelists are pointing out, the more we know about the virus, about the relation or correlation between host and pathogen, does bring us a bit more closer to understanding as to how the virus behaves, how we can fight it better. But as far as mutations of SARS-CoV-2 virus are concerned, there's nothing to worry about as of now, based on the knowledge we have, specifically in terms of the efficacy and safety of the vaccine candidates, which are being worked upon. But meanwhile, while the scientists are working on all these aspects, there are a few things which people like you and me can focus on, and that is ensuring a COVID-appropriate behavior. And therein comes three basic aspects: wearing a face mask, washing your hands regularly, and ensuring that you maintain physical distancing of at least 6 feet. All this done together will definitely see us through this crucial pandemic period, and we will be able to go ahead and avoid infection from COVID-19. We'll come back again with a different topic. Till then, keep watching Rajya Sabha Television. Thank you.